Hi all, welcome back. Um, we're continuing to talk about sensory and the sense organs or the sense systems. Last time out we were talking about hearing, and part of that hearing system that's directly related because it occurs in the same region is equilibrium. And so when we talk about equilibrium or your placement and how you adjust for where you're at kind of on the planet, whether you're laying down, standing up, hunched over, okay, kind of that gyroscope. If you think of a plane and the motion of that gyroscope, where it is relative to the longitudinal plane and the latitude plane for that matter, is really based on a couple sensories that we have, and we'll talk about both of them here. You have one that we're talking about when we talk about static equilibrium. Okay? This is really the position of the body and the head when you're not moving. Okay? So this has to do with gravity forcing down on your body and that kind of general position, and we'll look at the structure and how you can associate that. Okay? There are receptors found in the vestibula of the inner ear that are associated with this, and we'll cover these in a little more detail. Then you have what we call dynamic equilibrium. This is when you're actually moving. Okay, so movement of the head or the body, and this is what keeps you from toppling over. This is what keeps you and gives you the ability to kind of balance, to determine if you're moving too far to the left or too far to the right, and correct for that. And those are receptors that are found in the semicirculars of the canals. Okay, and so let's look at both of these, and they work very similar, and they have a very similar function. And in fact, when we look across lots of animals have very similar sensory to this, because all organisms that are moving have bodies that can move through the landscape, whether we're talking about a human, a frog, a bat, a bird, a fish. They all need to know where they're at on the planet, and it just happens to be that all of them have these kind of equilibrium sensory organs. So let's look at them. So if we look at that kind of labyrinth, um, you can find the maculae, which is this little sac-like structure. Okay? And if you dive into that maculae, you're going to see that there are hair cells associated with what we call otoliths. Otoliths just means like hard stone. Sometimes these can be calcium carbonate in the case of us. Other organisms, otoliths are actually bone. Okay, but regardless, you have all these little chunks of material, okay, that are inside this sac, this maculae, and this is kind of a semi-fluid material. Okay, it's got a little viscosity to it, but it flows. If you tilt your head or if you move, that material will flow and move. And when it does, it causes the hairs on the hair cell to move. And that stimulates the neurons to tell the position of the head. So if you move your head forward, bend it down, gravity pushes down on this gelatinous sac. Those otoliths move, and when they move, they stimulate the hair cells, and then that signal is sent. Same thing with going backwards, okay, etc. Okay. Now, if we're looking at dynamic equilibrium, okay, so this is moving your body and your head, kind of shaking. This is a little more complex and a little better from the perspective of not just knowing if you're moving down or up, but like, are you moving at an angle? okay? And that's really because of the labyrinths. So these labyrinths are full of semi-liquid, okay? And along those labyrinths, each one of the labyrinths, you can see that they move in different directions. There is a group of hair cells, just like the static, okay? And they, instead of being kind of encapsulated by a fluid-filled sac with bone or calcium carbonate with otoliths in it, they instead have a cupula, which is like this fluid, semi-fluid sac that encases the hair cells. And when you move, that cupula moves. And when that cupula moves, it moves the hair cells. And so you can see if you're moving in a certain direction, your head's rotating, that sac, that cupula will rotate, okay, will move in a direction based on gravity, based on flow, based on movement. That triggers signals, sensory neurons, to then tell the brain which direction the head is moving or which direction the body's moving. Again, the body's not really moving without the head, but you can trick all of these into doing very strange things by changing the composition of the fluid inside the sac. One way that we can do this, right, is by drinking large amounts of alcohol. And if you drink large amounts of alcohol, you change the fluid dynamic in the cupula, okay, which makes it thinner, and then tiny little movements, okay, will cause the solution in here to kind of continuously move. And for those of you who have experienced this, many of you probably have, okay, you get the spins, okay? And that's because your body can't tell its position. So it thinks that you're moving even though you're not, because the fluid inside the sac is moving. We can also do this by continuously spinning around in circles. Okay, so get on a merry-go-round or something like that, and you just continuously spin in circles, you become dizzy. Okay, that's a neuron or nerve response for the inability to know which way your body's moving, and that really has to do with tricking the system and stimulating these hairs in many different ways. Okay. All right, let's move to vision a little bit. Okay, so part of determining also equilibrium is vision. So of course some individuals, their vision like corrects for anything that has to do with the spins. Okay, so they might be able to go on a merry-go-round or spin in circles extremely fast, get off, and they may not be dizzy at all because their eyes can tell their brain, I'm on the right plane, I'm moving in the right direction, etc. Okay? But if you close their eyes or blindfold them, they may become extremely dizzy because their eyes don't have that capability of overcorrecting for the equilibrium that's happening in their ears. Okay, so anyways, back to vision. We can talk more about how you can trick your sensories, but vision in the sense is just sensing light waves, okay, light patterns. Now, in order to do that, we have to have different structures that are associated with your eyes. Okay, so you have, excuse me, upper and lower eyelids called the palpebrae. Of course, these provide protection. Eyelashes, which provide protection also. You have lacrimal apparatus, which provides tear production, which keeps these membranes, which are very thin, okay, and these thin membranes need to be moist, otherwise the cells will start to get damaged, and so you need kind of a tear production, moisturizer, okay? And then you have a series of different types of muscles that are attached to the eye so the eye can move in different directions, and those are extrinsic eye muscles. So let's look at a couple of these real quick. So the eyelids or the palpebrae. Okay? They're the thinnest membrane or the thinnest skin on the body. Okay? They are associated with muscles. Of course, you need muscles in order to move those gaps of limbs. The oculus oculi closes the eyelid, and the levator, okay, opens it. And so there's also lots of connective tissue, which help with uh, moisture, helps with energy, helps with providing space and structure to the eye. Okay, we'll talk about some of these. Some of these are glandular, so you can excrete like oils and other things that protect the eye and the eyelashes. Uh, so you also have mucous membranes that are associated with the eye that cover the portion of the eyeball. And if we look at some of these, okay, you can see you have a lot of subcutaneous or fat layer that's associated with certain regions of the eye. You have muscles, levator muscles, which can close or open the eye, sorry, and which are going to be associated with the eyelid. Okay. And then you have these different glands that are associated with the eyelid itself. Okay. The tarsal gland, which can excrete oil-like material that kind of provides protection. And a lot of that protection will be protection from not just abrasion. Of course, we need protection from abrasion because the eye itself is fragile, that surface is fragile, but also protection from things that are coming from the environment that might want to penetrate the body at that point. Okay, so bacteria, viruses. Okay, so sometimes these glands are excreting not just kind of oils that are high in fat, but they're also often will have some type of enzyme associated with it that would decrease the amount of bacteria that could get established in that region, that moist region of the eye. Okay, so we haven't talked too much about bacterial infections, but bacteria are normally seeking out moist, warm regions, and that's good breeding environment. If there's energy in the region, that's a good breeding environment for bacteria. It's a moist, warm region. The eye happens to be one, so you can get bacterial infections, but you have a few glands that are associated with it and the tear ducts that are associated with it that secrete enzymes that destroy bacteria and therefore viruses and fungi also. Okay, the lacrimal apparatus, the lacrimal gland. Okay, so again, this secretes tears. Tears are important for protection of the eye. Okay, these are associated or connected. The superior inferior canalicula are connected to the lacrimal sac and the nasolacrimal duct. And so this is why if you get, punched in the nose or a ball hits you in the nose or something like that, your eye ducts or your tear ducts start to release, even though, maybe from a pain perspective, it's not enough to be crying, but it's hit these sacs and ducts, and it's caused enough pressure that it releases tear ducts. It releases tears from the different glands. Okay, so the canaliculi are two ducts that collect tears. A lacrimal sac collects tears from that and lies in the groove of the lacrimal bone. Okay. And then the nasolacrimal duct collects the lacrimal sac and empties tears into the nasal cavity. Okay, so again, you can have release of tears. Tears can get collected, tears can leak out, okay, through the tear ducts, but they can also run out into the nasal cavity, either into the nasal passage and out the nose, Okay, so when you get a bad cold and you have lots of your ducts are releasing lots and lots of liquids, okay, you can get a runny nose, and part of that runny nose is going to be from tear duct, but also, you know, this is just a good way to get material that might have already went across the eye and removed kind of material to go through and out of the body. So it can go out the nose, but often it goes down the throat, and then out the body that way. Okay. So lysosomes or lysozymes, okay, again, these are enzymes that are antibacterial, which therefore become antiviral. Viruses have to have bacteria in order to reproduce. So if you get rid of the bacteria, you don't have viruses. But these are just really important enzymes that kind of wash the eye, protect the eye. They protect the different glands also. And just provide that protection, because again, this is a moist, thin membrane that would normally breed lots of bacteria, or is a great place for bacteria to breed. Okay, muscles that are associated with it. Okay, different muscles do different things. If we've kind of talked about, again, remember the purpose of a muscle is kind of to contract and then relax. So the contraction would cause the eye to move in different directions. Okay, so the superior rectus is going to erect, okay, or cause the eye to tip up. Okay, so things like the lateral rectus would move the eye to the side. Okay, inferior oblique moves the eye kind of at an angle in lots of different ways. And then, of course, you have muscles that are attached to the eyelid, which allows for them to close and open. And we already talked about those. Okay, so superior rotates the eye up, rotates the eye down as inferior. Medial rectus rotates the eye medially. Lateral rotates the eye lateral. Okay, superior oblique, eyes and down, rotates the eye down and lateral. Inferior oblique rotates the eye up and lateral. Okay, so this gives us the ability to move the eye in many different directions. And so, of course, it's not an all or nothing on these muscles. Okay, so you can have a little bit of a contraction, okay, so slightly up and to maybe to the media or towards the middle, slightly up towards lateral. So it's not a matter of all or nothing, and that's again stimulated by the nervous system, and it's controlled by you. And so those are skeletal nerves that are associated with those skeletal muscle. Let's go further into the eye. Okay, so we can look at the eye in general. It's going to be a hollow spherical organ. There are three main layers to the eye. You have the outer or the fibrous layer, okay, the middle, the vascular layer, and the inner, which is the nervous layer. Okay, so we have a specialized nerve, which we call the optic nerve, which is associated with the eye. Okay. So let's look at that outer layer first. Okay, the fibrous layer. Okay, this consists of the cornea and the scapula, and then portions of the outer tunic. And so if we look at the cornea, so this is this kind of region right here. This is what we're talking, this outer region. You have the cornea, okay, and then the sclera is right associated with that, okay, which is going to be on the back side of the eye. So if you follow that cornea around, okay, the sclera is going to be on that the back side of the eye. Okay. And then you have the tunic, which is going to be consist of a few different pieces. Okay, the cornea again is this outermost layer. This is kind of the protection layer. This is a layer that if you had a bacterial infection, this is where that bacterial infection is probably going to be established, is on that cornea. This is a site of this is the part of the eye that is associated with the external environment. Okay, so it's a transparent window. Okay, so it allows light to go through. Okay, and in some cases, just the structure itself helps focus light, or helps uh, helps light penetrate and be more condensed, especially in low light situations. Okay, it helps as many light or photons, waves, to make it into the lens as possible. Okay, and then that sclera, it protects the eye. It's tough, and tends to kind of be opaque, not quite see-through. Okay, so the cornea itself is transparent, but then when you move away from it, it's more opaque, and tough. It protects the eye. It's also where all the muscles kind of attach, and it's only pierced really by blood vessels and nerves. And we'll get into that a little bit later. Okay, kind of the setup of the eye. Okay, the middle, the vascular part, this is where a lot of the functionality of the eye occurs. And so this is where you're going to have specialized cells that occur in this region, like the choroid coat. Okay, so this is where you're going to have the blood supply, melanocytes, melanin. Okay, this is going to absorb the extra light that is not used for sensory purposes, okay, with the cones and the rods. And then you have cellular bodies, okay, these are pigmented, okay, that forms the ring that holds the lens, okay, and it also is associated with muscles that can move the lens forward, backwards, rotating, etc. And we'll look at this here in a second. Okay, and then of course the iris, which is the pigmented part of the eye, and that controls the light that's entering the eye. So you can think of it, iris, as a diaphragm, has the ability to open and close or change its aperture, okay, which allows more light to come in, less light to go in. So it really controls how much light is entering the eye. The cavity of the eye, okay, is the region between the cornea and the lens. Okay, this is liquid filled, which we call the aqueous humor. And the lens itself is biconvex, and that really allows for the light to be concentrated, and it focuses the light, concentrates the light into kind of a single beam. Okay, the more convex that is, the more concentrated the light is. So if it flexes and it becomes less convex, okay, it lets more light in, but it's not as clear of image. Okay, it's more convex, then the image becomes more clear, and but less light is let in. Okay. So we can accommodate that lens through what we call accommodations, okay, or change the shape of the lens to view different objects. Okay, so the lens becomes thicker, or more convex when focusing on close objects. Lens thins out and becomes flattened when you focus on distant objects. Okay, so again, you're looking at something and you're looking at fine detail. You need a single beam, basically a concentrated beam of reflectant. So everything you see is reflection. Okay, so you need that reflected beam that's coming off to be as tight a beam as possible when you're trying to look at fine detail. When you're trying to look at a wide atmosphere, you need as many photon wavelengths coming into the eye, so you kind of flatten the lens out so you can focus on distant objects. Okay, and you have a series of muscles and ligaments that are associated with that. Okay, so let's kind of look at this a little bit. So you have that cornea, you have the anterior chamber, you have the lens, and then you have associated muscles. So all these muscles that are associated with the lens, allowing for the lens to flex, bend, okay, become smaller, more elongated, etc. And then, of course, kind of already talked about kind of the pigmented region, the iris. Okay, that's all. Those are all associated with those muscles and ligaments that are associated with the lens that controls the structure, the portion of the lens that's movable. Okay, so iris itself can control the amount of light that enters the eye. Okay, and again, there's more muscles that are associated with the portion of the eye that allows for the eye's pupil to open or close. And so, again, pretty self-explanatory. Under dim light, okay, you need as much light to come in, so when it gets darker, your pupils will dilate. It means the pupil gets larger to allow as much light in as possible. When it's really bright, your pupil constricts, okay, because you don't need as much light. And a part of that constriction really has to do with melanin. So melanin is absorbing extra light, and it can only absorb so much, and this is why you'll get like white spots and things like that when you get really bright light. It's because the melanin can't pick up the extra photons that are coming in. But melanin is really what determines the eye color. That's determined by your genetics, by multiple genes will control it. And so you can have very different shades of color in your iris. Okay, the aqueous humor, again, this is the fluid that fills the eye, and it allows for the eye to, at least the eye to kind of be cushioned a little bit, but also it allows for nutrients and kind of ions, other things to be associated kind of that with the eyes. Of course, if you have any muscles in that region, which you got lots of them, they all need energy, and they all need sodium, they all need potassium, they all need calcium. And so these fluid filled areas are places where you can hold calcium and sodium and potassium, and you can have an excess amount of glucose that's in the region. And it's just a really good place for the transfer of material between different regions of the eye. Okay, so you can see that aqueous humor again, associated with the cornea. Between the cornea and the lens, you have kind of this liquid-filled region, chamber, that has direct connection with blood vessels. So you can get material coming in to the aqueous chamber, leaving the aqueous chamber, etc. And so that really allows for not just protection, but separation from the cornea to the lens. But it also allows for kind of the ability for that cornea to be flexed on without causing damage to the lens. Okay, the nervous tunic or the inner system. This is where your photoreceptors are. So their job is to pick up the photons that make it through the cornea and the lens, and then into kind of the back region of the eye. Okay. Few different regions. The macula lutea, okay, is the yellow spot in the retina. The photic centralis, this is kind of directly associated with the optic nerve, okay, and so that produces the sharpest vision. And then you have the optic disc. This is a region where there's a blind spot. Okay, so in most animals, we have a blind spot. We have a region where there's no photoreceptors. Right? And so you have nerve fibers that are leaving the eye in that region, and that really causes a region of blank or no photoreception. And I'll show it to you here on this diagram. Okay, so there's lots of layers. Of course, we'll dive into the photoreceptors here in a second. But the main purpose of this region is to take the stimuli from the wavelengths of photons. Okay, so photons move at different wavelengths, move at kind of different sizes. And those different sizes give off different energy. And that energy is then transformed in many cases to visual light cues. For us, those visual light cues are different color patterns, and that's what's being transmitted by the photoreceptors. Not just color, but also dark and light. But so these photoreceptors that we'll cover here in a second, okay, they're going to trigger kind of that impulse that we've talked about before that's needed by any of the sensory molecules. They're going to pick up some kind of sense. That triggers calcium gates to open. That can trigger sodium potassium channels to change. Triggers an action potential. That action potential moves down the axon and sends a signal to different regions of the central nervous system. In this case, the optic lobe of the central nervous system. Okay, so here's what that looks like. So you have light that's coming in. Of course, the light is going to move through the cornea to the lens. The lens is going to kind of restrict the light or cause the light to be bent, depending on the degree of bend or convex that the lens is will determine how intense the light is and where it would end up on the back of the wall here, the retina pigment. So the light's coming through. It has to travel past all these nerve fibers. This is one of the reasons why that the human eye is not super well designed compared to other organisms, because our light has to move through and go past neurons before it can get to the receptors. So it's going to get to the receptors. You have two types of receptors, cones and rods. Okay, so rods are sensing things like black and white or intensity of light, that kind of thing. And then cones are associated with colors. And we are what we call trichromatic. So we have three different types of cones to pick up three main different types of color, or three main different types of wavelengths. Okay. But anyways, the light will come through, associate with these receptor molecules, photoreceptor molecules. They'll send a signal. That signal then is sent back, okay, and then it'll go into the nerve fibers and then sent to the optic nerve and to the brain for interpretation. Okay, so also associated with all this region is a bunch of blood vessels. Okay, so not only does the light have to travel through the nerves, it also has to travel through the blood vessels. Okay, and those blood vessels, they come through in one region, the optic disc. Okay, and that's going to be where the blind spot of the eye is. Okay, so this region right here is going to be a region where there is poor vision, actually no vision. Okay, and then you have regions where there might be less blood vessels, less nerves, like the forae centralis, Okay, that's going to be the spot that has the best vision, the best focal point.